Good day, my name is Jomar F. Caming from BS Fisheries 3, and this is my preliminary examination of fish genetics and breeding. Number 1 question: define genetics and its importance in fisheries. Genetics is a study of heredity and the variation of inherited characteristics of unliving things. In fisheries, genetics have been used to produce monosex population, sex reversal, YY technology, and hybridization. Production of new strains to produce fast-growing and disease-resistant strain to increase production per unit area. It is also used in determining population size and intraspecific relationship among species. In stock enhancement, conservation biology, and it's also a legal application. Number 2, what is the chromosomal and chemical basis of heredity? The chromosomal theory of inheritance was given by Boveri and Sutton in the early 1900s. It is the fundamental theory of genetics. According to this theory, genes are the units of heredity and are found in chromosomes. Chromosomal theory of inheritance came into existence long after Mendelian genetics, and the chemical basis of heredity are deoxyribonucleic acid is the chemical basis of heredity in organisms. DNA is present inside the nucleus. It contains genetic information instruction for protein synthesis. It passes down from parents to offspring. Number 3, explain the concept of population genetics. Population genetics is the study of genetics variation within among population and evolutionary factors that explain this variation. Its foundation is the Hardy-Weinberg law, which is maintained as long as population size is large, mating is at random, and mutation, selection, and migration are negligible. And number 4, what is the importance of selection in fish stock management? The purpose of stock management assessment is to provide information to fishery managers that will allow them to control the catch of its species or species complex so that ideally its population can be maintained to produce. And number 5, differentiate hybrids from inbreeding and plan a good crossbreeding program. Inbreeding is the production of offspring by related parents. The most extreme form of inbreeding is self-fertilization, which is possible in a few species of scallop. Systematic pattern of mating, such as pulsive matings, habsive matings, all result in inbreeding. In population of finite size, inbreeding accumulates over time, even with random mating, as by chance some relative individuals will be mated in contrast to inbreeding, which increase the frequency of homozygotes crossbreeding, or the mating of unrelated individuals increase. By increasing the frequency of heterozygous genotypes, recessive alleles are less likely to be expressed, and so the fitness of the population can be increased for one generation following crossing. At least increased heterozygosity of crossbreed individuals is often observed as hybrid vigor or heterosis, when the average value of the offspring for the particular trait exceed the mean of the average values of the two parental lines. And number 6, identify biotechnologies in fish genetics and determine how could help increase productivity. The common methods used in fish biotechnology today are chromosomes modification and hormones treatment, which can be used to create triploid, tetraploid, haploid, gynogenetic, and androgenetic fish. Because of the world's rapid population growth, there is a growing demand for fish, and many natural freshwater and the ocean fisheries have reached their harvest limit. It is unlikely that these demands can be met by higher natural harvest. Number 7. Differentiate mitosis from meiosis. Mitosis is the process where the single cell divides into two identical daughter cell division, and meiosis is the process where the single cell divides twice to produce 4 cells containing half the original amount of genetic information. Number 8. Differentiate DNA from RNA. DNA is a double-stranded molecule that has a long chain of nucleotides. DNA replicates on its own. It's self-replicating. RNA is a single-stranded molecule which has a shorter chain of nucleotides. RNA does not replicate on its own. Number 9. Discuss how and why DNA replicates. The DNA is copied or replicated once per cell division. It's copied every cell division within the lifetime of one organism, and its reproductive cell line that produce sperm and eggs. A large set of perhaps 50 or so enzyme catalyze the DNA replication. DNA polymerases are the most important of this enzyme. And number 10, explain the importance of transcription and translation. The process of transcription and translation act to decode the DNA information in gene. The result is the protein molecule, and it's through protein that DNA exerts its effect in the cell of living creature. Number 11, discuss how proteins and amino acid are synthesized. Amino acids are synthesized from intermediates in glycolysis, the citric acid cycle, or the pentose phosphate pathway. Nitrogen is provided by glutamate and glutamine. Amino acid synthesis depends on the formation of the appropriate alpha keto acid, which is then transaminated to form an amino acid. Number 12, explain the principle of YY male technology and the process of how to produce YY males. The YY male tilapia or fish technology is based on the genetics manipulation of sex. This is achieved through a combination of feminization and progeny testing to identify the novel YY genotype that sires only XY natural male natural male progeny or natural male tilapia. Number 13, explain the different kind of artificial selection methods: mass selection, family selection, and multi-trait selection. Mass selection, superior individual of the best families are maintained for breeding progeny testing males of the best families. Family selection is the strategy whereby the breeding candidates are ranked based on records taken on their full and half sibs, that is individual that have genes in common with them. The selection in which several traits are improved in succession, such as selection is carried out fish for one trait until the desired genetic level is reached, then selection for the second, third, and the following traits are performed. And number 14, advantage of crossbreeding. Ito nung tinatawag natin na advantage ng crossbreeding natin. Crossbreeding is the opportunity to capitalize on breed complementarity. This involves evaluating the strengths and weaknesses of potential breeds and selecting those that complement each other. The result should be an animal that has the best traits of those breeds. Thank you and good day.